Good morning, everyone. Today we will start with a new topic of class 11th biology. The name of the topic is muscular tissue. It is a part of chapter animal tissues.
In this video, we will study about muscular tissue, various types of muscular tissue, and sliding filament theory.
Muscular tissue is a specialized tissue in animals which applies force to different parts of the body by contraction. It is made up of thin and elongated cells that are called as muscle fibers. It controls movement of an organism. Muscles are made up of highly specialized thin and elongated cells which are called as muscle fibers. These muscle fibers contain specialized cytoplasm, and that cytoplasm is called as sarcoplasm, which contains network of membrane called sarcoplasmic reticulum. It is actually the endoplasmic reticulum of muscle cells. The muscle fibers may be bounded by the cell membrane called sarcolemma. Each muscle fiber contains numerous longitudinal fibrils
Called myofibrils.
Muscles are specialized tissue of mesodermal origin and made up of contractile fibers which in turn are formed of myofibrils. In human adult, muscles constitute nearly 40 to 50% of the total body weight. Muscle has four major functional characteristics that includes contractibility, excitability, extensibility and elasticity. Contractibility is the capacity of muscle to contract or shorten forcefully. Extensibility is the ability of muscle to be stretched to their normal resting length and beyond to a limited degree. Elasticity is the ability of muscle to recoil its original resting length after it has been stretched. Excitability is the capacity of muscle to respond to stimulation by nerves and hormones, making it possible for the nervous system and, in some muscle types, the endocrine system to regulate muscle activity. Now we'll discuss the basic structure of muscular tissue. The muscular tissue are bundled together and are surrounded by a tough connective tissue similar to cartilage known as epimysium. The bundle of nerve cells that run in long fiber called fascicles are surrounded by epimysium. The fascicles are surrounded by a protective layer known as perimysium. It allows the flow of nerves and blood to the individual fiber. Another protective layer, the endomysium, surrounds the fibers. These layers and muscles helps in contraction of different parts of the muscles. The different bundles slide past one another as they contract. The epimysium connects to the tendons attached to the periosteum, connective tissue that surrounds the bone. This helps in the movement of skeleton when the muscles contract. The epimysium connects to the other connective tissue to produce a force on the organs and control everything from circulation to food processing. Based on the location, in humans, muscles are broadly classified into three categories: skeletal muscles, smooth muscles, and cardiac muscles. Skeletal muscles. Skeletal muscles are the muscles which are attached to the bones by tendons and help in the movement of the parts of skeleton. As these muscles are under control of conscious mind and can be moved according to our own will, these are also termed as voluntary muscles. Skeletal muscles are innervated by voluntary nervous system. Under the microscope, skeletal muscles exhibits transverse strips, hence they are called as striated muscles. The muscles are responsible for locomotory action and change in body posture. Each skeletal muscle fiber is a single cylindrical muscle cell. An individual skeletal muscle may be made up of hundreds or even thousands of muscle fibers bundled together and are wrapped in a connective tissue covering. Each muscle is surrounded by a connective tissue sheath called epimysium. Fascia, a connective tissue outside the epimysium, surrounds and separates the muscles. Portion of epimysium projects inward to divide the muscle into compartments. Each compartment contains a bundle of muscle fibers. Each bundle of muscle fiber is called fasciculus and is surrounded by a connective tissue called perimysium. Within the fasciculus, each individual muscle cell, called a muscle fiber, is surrounded by a connective tissue called endomysium. Skeletal muscle cells, like other body cells, are soft and fragile. The connective tissue coverings furnish support and protection for the delicate cells and allow them to withstand the forces of contraction. The covering also provides pathway for the passage of blood vessels and nerves. Commonly, the epimysium, perimysium, and endomysium extends beyond the fleshy part of the muscle, the belly or gaster, to form a thick rope-like tendon or a broad flat sheet like aponeurosis. The tendon and aponeurosis forms an indirect attachment from the muscles to the periosteum of the bones or to the connective tissue of the other muscles. Typically, a muscle spans a joint and is attached to a bone by tendons at both the ends. One of the bones remains relatively fixed or stable while the other end moves as a result of muscle contraction. Skeletal muscles have abundant supply of blood vessels and nerves. This is directly related to the primary function of the skeletal muscle, that is contraction. Before a skeletal muscle fiber can contract, it has to receive an impulse from a nerve cell. Generally, an artery and at least one vein accompany each nerve cells that penetrates the epimysium of the skeletal muscle. Branches of the nerves and blood vessels follows the connective tissue components of the muscles of a nerve cell and with one or more minute blood vessels called capillaries. Because skeletal muscle cells are long and cylindrical, they are commonly referred to as muscle fibers. Skeletal muscle fibers can be quite large as compared to that of other body cells. Having many nuclei allows the production of large amount of proteins and enzymes needed for maintaining normal function of these large protein dense cells. In addition to nuclei, skeletal muscle fibers also contains cell organelles found in other cells such as mitochondria, endoplasmic reticulum, etc. However, some of these structures are specialized in muscle fibers. These specialized smooth endoplasmic reticulum, called sarcoplasmic reticulum, stores, releases, and retrieves calcium ions. The plasma membrane of muscle fiber is called sarcolemma, and the cytoplasm is referred to as sarcoplasm. Within a muscle fiber, proteins are organized into structures called myofibrils that runs the length of the cell and contains sarcomeres connected in series. Because myofibrils are only approximately 1.2 micrometer in diameter, hundreds to thousands can be found inside one muscle fiber. A sarcomere is defined as the region of myofilament contained between two cytoskeletal structures called Z discs, and the striated appearance of the skeletal muscle fiber is due to the arrangement of the thick and thin myofilaments within each sarcomere. The dark striated A band is composed of the thick filaments contained myosin, which spans the center of the sarcomere extending towards the Z disc. The thick filament are anchored at the middle line, that is called as M line, by a protein called myomycin. The lighter I band region contains thin actin filaments anchored at the Z disc by a protein called alpha actinin. The thin filament extends to the A band towards the M line and overlap with the region of the thick filament. The A band is dark because of the thicker myosin filament as well as overlapping with the actin filaments. The H zone is the middle of the A band is a little lighter in color because the thin filaments do not extend into this region. Because a sarcomere is defined by Z disc, a single sarcomere contains a dark A band with half of the lighter band on each end. During contraction, the myofilaments themselves do not change length but actually slide across each other. So, the distance between the Z disc shortens. The length of A band does not change. Myosin filament remains a constant length, with the H zone and I zone region shrinks. These regions represents the area where filaments do not overlap. Structure of contractile protein. The thick myofilaments are formed of myosin proteins, whereas the thin myofilaments are formed of three types of proteins called actin, troponin, and tropomyosin. These four proteins are collectively known as contractile proteins. Thick myofilament or primary myofilament. It consists mainly of myosin protein. Each myosin filament is a polymerized protein made up of many monomeric proteins called meromyosins. Each meromyosin has two important parts as a globular head and tail. A globular head has a short arm called heavy meromyosin and a long arm called light meromyosin. The heavy meromyosin components projects outward at a regular distance and angle from each other and from the surface of a polymerized myosin filament and are known as cross arms. The globular head is an active ATPase enzyme which has binding sites for ATP and active sites for actin. This tail is called the light meromyosin. The myosin molecule has two identical heavy chains and four light chains. The two identical heavy chains wrap spirally around each other to form a double helix. The light chains are the part of myosin heads and help to control the function of head during the contraction of muscles. Thick filament or secondary myofilament. It is composed of three proteins. The first one is actin protein, which is a globular protein with a low molecular weight. It is made up of two filamentous actins called as F-actins that are helically wound around each other. Each F-actin is a polymer of monomeric subunits that are called as globular actins or G-actins. The two filaments of tropomyosin protein runs close to the F-actins throughout the length. The troponin is a complex of proteins of three globular peptides that includes troponin T, troponin I, and troponin C that are distributed at a regular interval on tropomyosin. In the resting stage of a muscle fiber, a subunit of troponin masks the active sites of myosin on the actin filaments. Partial overlapping of primary myofilament by the secondary myofilament imparts dark appearance to the A band. The strong affinity of troponin for the calcium ion is believed to initiate the contraction process. Now let's discuss the mechanism of muscle contraction. The contraction of muscle is best explained by the sliding filament theory. It states that contraction of muscles takes place by the sliding of thin and thick filaments that passed over each other with the help of cross bridges to reduce the length of the sarcomere. This theory was proposed independently by A.F. Huxley and R. Niedergerke and by H. E. Huxley and Jean Manson in England in 1954. The sequence of events leading to the contraction is initiated by a signal in the central nervous system, either from the brain or from the spinal cord via motor neuron. A motor neuron, along with the muscle fiber connected to it, forms a motor unit, and the action potential is conveyed to the motor end plate. That is the junction between a motor neuron and a sarcolemma of the muscle fiber on each muscle. A neurotransmitter is released at the junction by the neuron signal, which generates an action potential in the sarcolemma. This spreads and causes the release of calcium ions into sarcoplasm.
Calcium ions plays a key regulatory role in the muscle contraction.
Increase in calcium ions level leads to their binding to troponin subunit, thus exposing the active sites on F-actin molecules. An ATP molecule joins the active site on the myosin head of myosin myofilament. These heads contains an enzyme myosin ATPase that along with calcium ions and magnesium ions catalyzes the breakdown of adenosine triphosphate. The energy is transferred to myosin head which energizes and strengthens to join an active site on actin myofilament forming a cross bridge. The energized cross bridges move, causing the attached actin filaments to move towards the center of A band. The Z line is also pulled inward, causing shortening of sarcomere, that is, contraction. It is clear from the above explanation that during contraction, A bands retain the length while I bands get reduced. The myosin head releases ADP and inorganic phosphate, relaxes to its low energy state. The head detaches from actin myofilaments when new ATP joins it. In repeating cycle, the free head cleaves the new ATP. The cycles of cross bridge formation and breakage is repeated causing further sliding. After contraction, the calcium ions are pumped back to the sarcoplasmic cisternae, blocking the active sites on actin myofilaments. The Z line returns to its original position, that is, relaxation. Rigor mortis is the state of body stiffening after death due to non-separation of actin and myosin filaments caused by the non-availability of ATP. It appears first in the small muscles of the face, that includes jaw, and those being used most actively prior to death. It persists till decomposition starts. Skeletal muscles undergo powerful and rapid contractions with short rest periods and hence get fatigued very easily. They are supplied by voluntary nervous system that includes central nervous system and peripheral nervous system. They require large amount of energy as they have to help in movement and locomotion. So are supplied with blood vessels and numerous elongated mitochondria and glycogen granules. They are found attached to head, trunk, limbs, also in body walls, tongue, pharynx and esophagus. As they are voluntary in function, they bring about the movement of body organs and locomotion of the body. Types of skeletal muscles. There are two types of skeletal muscles that are named as red muscles and white muscles. Red muscles are due to the red pigment called myoglobin, which is in high amount in human body. These muscles are smaller in diameter and have a large number of mitochondria in it. The myoglobin stores the oxygen which is used by the mitochondria for the synthesis of adenosine triphosphate. Red muscles have a large number of blood capillaries in it. Next is white muscles. Unlike the red muscles, the white muscles are bigger in diameter and they have a small amount of myoglobin in it. They also have less number of mitochondria in it. Functions of skeletal muscles. There are a number of important functions that are performed by skeletal muscles. As these muscles are in our own conscious control, so these muscles are responsible for the body movements such as typing, breathing, extending the arm, writing, etc. The muscle contract, which pulls the tendons on the bones and causes movement. The body posture is maintained by skeletal muscles. The gluteal muscle is responsible for the erect posture of the body. The sartorius muscle in thigh are responsible for body movement. The skeletal muscles protect the internal organs and tissues from any injury and also provides support to these delicate organs and tissues. These also supports the entry and exit points of the body. The sphincter muscles are present around the anus, mouth, the urinary tract. These muscles contract, which reduces the size of the openings and facilitates the swallowing of food, defecation,
and urination.
The skeletal muscles also regulates body temperature. After a strenuous exercise, the body feels hot. This is due to the contraction of skeletal muscles which converts energy into heat. Smooth muscles. Smooth muscle is so named because, unlike other forms of muscle, the arrangement of contractile proteins does not give the appearance of cross striations. Smooth muscles are the most widely distributed muscles in the body. They are also named as visceral muscles, as they form the muscular component of the visceral organs such as blood vessels, gastrointestinal tract, the uterus, urinary bladder, iris, ciliary body, etc. Smooth muscles are also known as involuntary muscles since smooth muscle is under automatic and hormonal control. Smooth muscle fiber is a spindle shaped with only a single central nucleus. There is no distinct sarcolemma but a very fine membrane surrounds each fiber. Smooth muscles never connect with skeleton. Actin and myosin myofilaments within the myofibrils are very thin and are arranged more randomly than in skeleton muscles. So there is no stripes. Smooth muscle cells do not have the troponin tropomyosin mechanism of controlling contraction. The calcium is bound to the calcium binding protein calmodulin, which acts as a second messenger. The calmodulin calcium complex activates an enzyme myosin kinase promoting contraction. There are two main characteristics of smooth muscles. Its contraction and relaxation period are slower. Its action is rhythmical. Its contraction may last for 30 seconds or more, but it does not tire easily. Such sustained contraction pulls the ability to stretch made it suitable to muscular control of stomach, intestine, urinary bladder and uterus. There are two main types of smooth muscles. Single unit smooth muscles, which are also called as visceral muscles. Here, all the cells function collectively and simultaneously as a single unit. They composed of muscle fibers closely joined together, which contract as a single unit. For example, the muscles of urinary bladder and gastrointestinal tract. The second one is multi-unit smooth muscle. It is a type of muscle in which all the cells cannot function collectively and works independently. For example, hair root muscles and the muscles on the large blood vessels.
Single unit smooth muscle. These cells forms the walls of all inner hollow organs of the body. It creates long and uniform contraction that facilitates the passage of substances through the body such as food digestion in the gastrointestinal tract. These muscles are also present in the blood vessels which integrate structures and controls the vessel diameter by contraction and relaxation of the vessel. It is a tissue associated with the organ present inside the body, especially those in the abdominal cavity. In other words, we can say that visceral muscles is found near or in the abdomen. Location of single unit smooth muscles. Smooth muscles are located in the internal lining of the different hollow organs like the reproductive system, urinary bladder, liver, and the circulatory system's blood vessels. Multi-unit smooth muscles. These muscles consist of cells that cannot function collectively and work independently. Location of multi-unit smooth muscles. Such muscles are mostly located in the lungs. The erector pili muscles associated with hair follicle in the large arteries and also in the internal eye muscles controlling the entry of light and lens formation. Functions of smooth muscles. The basic functions of smooth muscles in the organ system are as follows. In case of gastrointestinal tract, it helps in propulsion of food bolus. It also helps in the regulation of blood flow and pressure via vascular resistance in cardiovascular system. It also helps in regulation of urine flow. Contraction during pregnancy and propulsion of sperm is also regulated by smooth muscles. Regulation of bronchiole diameter is also regulated by smooth muscles. Smooth muscles raises hair with erector pili muscles. It helps in dilation and constriction of the pupil as well as the changing lens diameter. Cardiac muscles. It is present only in heart. If we talk about the structure of cardiac muscles, under microscope they have similar striations as skeletal muscles. Cardiac muscle cells are closely packed, but each cell are nucleated and separated from each other. The cells are joined end to end by the specialized cell junction called intercalated discs that attaches one cell to another with desmosomes. They connect the myofibril filaments of adjacent cells and contain gap junctions that helps to synchronize the contraction of smooth muscles by allowing impulse transmission from one cell to another. They contain light and dark bands, light I band and dark A band. The intercalated disc always occurs at the location of Z line. It is supplied with central and autonomous nervous system. The rhythmic contraction is its own, so they are voluntary in nature. They do not get fatigued easily, so they are called as fatigue-less muscles. Functions of cardiac muscles. Cardiac muscle tissue work to keep our heart pumping through involuntary movements. This is one feature that differentiates it from skeletal muscle tissue, which you can control. It does this through specialized cells called pacemaker cells. These control the contraction of our heart. Thank you everyone.